Assalamu alaikum. Let me welcome you to a presentation on cardiovascular system. This is the first part of the cardiovascular system presentation, and this presentation will be completed in two parts. In the first part, the general idea on the cardiovascular system, various types of circulation, general histological structure of the blood vessels, general idea on the changes in the wall of arteries, classification of the blood vessels, and the clinical correlations at all stages will be considered. You can see this one is a road, just a sketch of a road. Suppose we are interested to send this medical storage to a distant place. What we need? We need one transport, one media which will carry it to that place, and we need energy that will thrust this transport to that place. So for a circulation, one pathway is required. Suppose this is a tube. So if fluid wants to circulate, then needs a pathway that should be tubular. And suppose this rate drop is interested to circulate from one end to other, so we need one fluid media. Suppose this yellow stain is the fluid media. If we place a pump here, then a circulation will be created, a wave will be created in the fluid media, and that can carry it to a desired place. So for a circulation, we need one pressure, one pump, or one energy source. There should be a pathway, and there should be a transport who will carry the desired substance. So this is actually indicating the circulation. Why this circulation should be there? There is to take something to a place and to carry out something waste to a particular place. So this circulation will be helpful to carry required nutrition to the tissue and can drain waste product from the tissue to a place where this will be eliminated. So every time this circulation is to meet the demand of the cell, and different fluid media are continuously circulating to carry the substances to the cell. So this is circulation is to supply the required substance to a particular place and drain the waste substance from that place to a site where this will be separated from the transport media and will be eliminated from the body. This is the central theme or central requirement of circulation. In the body, we have blood circulatory system. The second important is the lymphatic circulatory system. Other than that, we have CSF circulatory system, then aqueous humor circulation, and of this, in this presentation, we will try to cover the blood circulatory system only. Blood circulatory system means this has blood and that should circulate through a tubular system, and this has some pump. So these are the components, and there are different types of circulation present in the human body. That may be shorter, this is lesser circulation, and maybe bigger, this is greater circulation. So this circuit which is smaller is lesser circuit, and this circle will carry blood from heart to the lung and return back to the heart, and the objective of this circulation is to have exchange of oxygen from the atmosphere to the blood, and carbon dioxide from the blood to the atmosphere. The greater circle, this is a larger one, and this loop passes through rest of the part of the body. Also it includes the lung. and this may be of two types: systemic circulation and portal circulation. Systemic circulation means it contains one capillary bed in the loop, and portal circulation, there are two capillary beds in each loop. So the number of the capillary bed is the factor to for determination of the type. You see, this is the diagram showing. So from the heart, the blood is going to the lung and it is oxygenated, carbon dioxide is eliminated, then it is returning back to the heart again. So this circulation is the pulmonary circulation, and it is lesser in structure. It is a smaller one. Now blood is from the heart going to different part of the body. This is going to the head neck region and making one capillary and returning back. Another going to the lower part of the body, making one capillary returning back. It's also through kidney, one capillary returning back. This is systemic circulation. If it goes through one set, then second set with different functions. Look at this is the pulmonary circulation. So you can see this is the right atrium. Blood enters into this from all over the body. From here the blood goes to the right ventricle through the atrioventricular opening, and from the ventricle it enters into the pulmonary trunk. Right ventricle to pulmonary trunk, this is the pathway and it's guarded by the valve. Then it divides into two and takes it to the left and right lung and divides into capillaries to get an exchange of atmospheric gas and the blood gas. And then it returns back to the left atrium. This circulation which starts from the right ventricle, passes through the lung and returns back to the left atrium, is pulmonary circulation. The function of this circulation is to oxygenate the blood. You see, this is the pulmonary circulation level here. This one is the portal circulation because it is having two sets of capillary, you can see here. And this is systemic circulation because it is having one capillary bed, the rest of the part of the body. And this is the heart. This one is the blood vessels and it contains the blood. So these are the component parts of the cardiovascular system or blood circulatory system. So we got an idea of general circulation as well as component of cardiovascular system. Blood vessels, anatomy of the blood vessels is very important to understand the activities of the cardiovascular system. So let us concentrate on the general anatomy of blood vessels. You see, these blood vessels are of three types: artery, then it divides into capillaries forms, and then capillary unites together to form the veins. So these are the arteries. We can define artery as the vessels who carries blood from the heart to the tissue. So it may carry oxygenated blood or deoxygenated blood. So nature of the blood is not important for defining the artery, capillary and the vein. These are the veins who carries blood from the tissue to the heart. So direction of the blood is important for the defining the vein and artery. Now these are the capillaries. These are very thin walled. Actually it comprises of simple squamous with very thin and connective tissue layer is the capillary. So endothelium lined tube, and this is for the exchange of the gas between atmosphere and the blood are the capillaries. Blood vessels are tube. So tube means it comprises of several layers and we can describe the histological structure by naming the layers and studying the content or composition of the layer. So blood vessels comprises of three layers. If we start from inside, the tunica intima, tunica media and tunica adventitia are the layers. So this is the tunica intima, inner part, then is the media, and after that outside is the adventitia. If we say this is the vein and this is the diagram of artery, there is some change in the nature of the blood vessel. So here you can see this is the intima and this bracket is indicating the media and this is adventitia. So this nature and structure of the layers varies from arteries to arteries and veins to vein. You can see a 3D appearance of the blood vessels and several layers are shown. The histological section shows its layer here. This is the inside outward, the tunica intima, this is the lumen and this is the lumen. So on the surface you can see this is the simple squamous epithelium, nucleus you can see, and a wavy elastic layer is there. This is in section, it is looking like line but it is a sheet, and you can see here also, and the nucleus are very close to this. So this endothelium and this lamella, this is known as internal elastic lamella, very close or may be separated by a layer of connective tissue. This connective tissue is called subendothelial connective tissue. So intima comprises of endothelium at all places, so everywhere it is simply squamous. Then it lies on a thin layer of connective tissue that varies in thickness, maybe a bit thicker or maybe very thin. After that we will get one sheet of elastic tissue and this is known as internal elastic lamina. So up to this is the intima. So lining epithelium is simple squamous, which is known as endothelium. After that, loose connective tissue known as subendothelial connective tissue or subendothelium. Then is the internal elastic lamella. This is a sheet of elastic type of connective tissue. Tunica media is this one. After the internal elastic lamella, this tunica media starts. So this is very thick tunica media. This is internal elastic lamella. After that, this one is the internal elastic lamella, and after that, this is the media. So media comprises of a concentrically arranged smooth muscle. These are the smooth. The spindle shaped nucleus is the smooth muscle. Its cell is also spindle shaped. Lower power view, you can see these are the nuclei and all are connective smooth muscle cells. They are circularly arranged. In between that there may be some elastic lamella, and the ratio of the muscle and the elastic layer varies from size to size of the artery and also from the vein. After that, outside you can see similar to the internal elastic lamella, one elastic lamella is there, but it is relatively less marked. This layer is known as external elastic lamella. So the external elastic lamella is part of media and internal elastic lamella part of intima. Now you can see the internal elastic lamella clean. This is media, and after that at the junction there is external elastic lamella, and outside that is the adventitia. Tunica adventitia is a connective tissue layer, and it surrounds the outer part, and in vein it is very thick, in artery relatively thinner, and this layer, if the total wall is thick, this layer contains some own blood vessel. You can see the histological section of artery, and this is the endothelium. After that you can see an elastic lamella, that is the red mark. Then media start, external elastic lamella, and this is the adventitia. So outside this is the connective tissue, adventitia is continuous with the connective tissue. So tunica media is variable layer that will help us to identify what type of artery is it, or is it vein or artery. So media is a very important clue to the determination of the blood vessels. Relative thickness of the wall is also an important factor. Media is thickest layer in the artery, but adventitia is the thickest layer in case of vein. This is the heart, and within heart how the blood circulates. So blood from all over the body enters into the right atrium. This is the place of the right atrium. Through the right atrioventricular opening, the blood enters into the right ventricle. From the right ventricle, through this opening that guarded by the valve, enters into the pulmonary trunk. So through this pulmonary trunk, the blood is distributed to the lung by the pulmonary artery. From the lung, after purification, means adding oxygen and removing carbon dioxide, the blood becomes pure and returns back to the right atrium of the heart. From the right atrium, it passes through the right atrioventricular opening to the right ventricle, and finally again this purified blood is pushed back to this aorta that lies behind the pulmonary trunk, and it carries blood to the different parts of the body. This is the ascending aorta, and this one is the arch of the aorta. So immediately a large amount of blood enters into the blood vessels, and ventricle pumps this into the blood vessel. So the blood vessel should dilate to accommodate this extra amount of blood. So the initial part, who are close to the heart and receiving blood, should have wall that is elastic in nature, so that it can dilate and receive blood. Now when it gets the blood, immediately the wall is stressed. So this, because of the elasticity, this will try to return back its original position. So there will be continuous elevation of the blood pressure. The highest pressure is known as systolic, and it tries to maintain the blood pressure and prevent the total fall of the blood pressure to the zero. So it is held at a particular pressure. This is diastolic pressure. How these blood vessels are changed from larger to gradual change. Already I have described that the initial part, who is receiving the blood, should have elastic wall. This is ascending aorta, and this should have elastic wall. Gradually the artery divides and have the wall different organization and different thickness, and ultimately become very thin capillaries. After that again it gets a wall that is relatively different in nature. So structure and thickness will give an indication of what type of blood vessel. So artery may be classified on direct observation. This is gross classification. Under microscope is the histological classification. So grossly, artery may be large artery, medium sized artery, then it becomes small artery and arteriole. Histologically, the wall composition is elastic in nature, then it becomes muscular in nature, and then a definite type of wall containing artery is known as arteriole, that we will describe detail. So this is the classification. Here you can see the medium and small, making this classification 4, but here it is 3. This medium and small artery is histologically muscular artery. Veins of the different size you will find, and they may be large, medium and small, and at the beginning, after the capillary, they become venule. So every structure we will discuss one by one. You can see this are the capillaries, so they drain into the venule. This is arteriole carrying capillary, then venule, and venule unites together to form small vein, then medium vein, then large vein. So let us summarize the classification. We got 3 types of classification. Histology means observed under microscope, then gross classification, which we can classify depending on our naked eye vision, and there is another type is the functional classification depending on the function of the blood vessel. So artery may be large artery which is histologically elastic in nature. They are called the conducting vessels, means they carry the blood towards the periphery. They conduct the blood to the medium and smaller size of blood vessel. This medium and small are muscular artery. Their wall contains smooth muscle. These are the vessels, that arteries that takes the blood towards some organs. That's why it is known as distributing vessels. Arterioles are grossly also known as arterioles. They have definite wall. I will discuss it in a subsequent slide, and they are responsible for maintaining a resistance, and this resistance enables to blood to maintain its pressure. Capillaries are histologically classified into continuous, fenestrated and sinusoidal capillaries, and these fenestrated may be with or without diaphragm. These capillaries are functionally known as exchange vessel. Veins histologically, usually we used to say venules with definite wall and veins as a common in nature. Grossly we used to say venules that unite together to form small veins, gradually becoming larger because of the union of multiple veins, and finally there are large veins. These veins are functionally known as reserve vessels. So conducting vessel distributes the blood to the organs and become narrower to maintain the pressure of the blood. Resistant vessels, there will be capillary for the exchange and return back, and with the low pressure, large amount of blood is remaining in the vein. That's why it is known as reserve vessel. About 20% blood remains in artery and 80% blood remains in the veins, means 4 times more blood than artery. A large amount of blood remains in the capillary too. If we compare the artery and vein, then vein contains 4 times more blood than the artery. This is a histological structure of the elastic artery. You can see these dots are on the, this is the lumen. So endothelium nucleus is dot. After that you can see a dark red color. This is internal elastic lamella. Then starts media. You see a tunica intima has endothelium. It is thick, thicker than other part of the blood vessels, but not than media. This is having endothelium, then is the subendothelial layer, and finally the internal elastic lamella. This is the media. You see so many elastic lamella are there. So actually this internal elastic lamella is very tough to distinguish from other elastic lamella. in larger artery. But we will see this is different in case of muscular artery. So tunica media is thickest layer of the three, and it contains smooth muscle as well as large amount of elastic lamella. This wavy lines are all elastic lamella. So this is full of smooth muscles and elastic lamella. Smooth muscle concentrically in between elastic lamella. Adventitia is relatively thinner, but it is the second most thick layer, and it comprises of dense connective tissue. You see, the blood vessel is very large, so it is containing some blood vessel in its wall. This is the muscular artery. This is the lumen. So inside you can see the endothelium. Intima is not that much thick as the elastic artery. If you look inside, you'll find very prominent internal elastic lamella. It's very prominent because media is not having such complete elastic lamella, which is present in the elastic type. So here the media is thick layer but contains many circular muscles, concentrically arranged muscles. So here intima is having prominent internal elastic lamella that can help you to distinguish it from the elastic artery, and here the media is thick and it is containing circularly oriented smooth muscles. Elastic component is very less and it reduces from media to smaller one. Tunica adventitia is the outer coat. It is the dense connective tissue layer. Here we can differentiate elastic artery from muscular artery. Look at the right one. The internal elastic lamella is very prominent. Here it is has merged with the elastic lamella of the media. Media on the muscular artery, a very few elastic layer you can see, but mainly smooth muscle is there. But here elastic layer is a very large component, and in between there are smooth muscle. This is the adventitia and here adventitia. So it is very tough to distinguish these two observing the adventitia. Media is the thickest layer, intima is thinner in case of muscular artery, relatively thicker in the elastic artery. So these are the distinguishing point between the elastic and. Now artery divides and divides, becomes smaller and smaller. So the layer decreases. Intima, you see, endothelium is unchangeable, and there is outside that connective tissue is there. So it reduces in size, but comparatively media reduces much. So media muscular layer reduces. Suppose 50 layers of muscles are there, that will be 40, then 30, and ultimately a few. When the muscular layer after endothelium, the media contains only 1 or 2 layers of circular smooth muscle. This is arteriole. So arteriole has media containing 2 layers of smooth muscle or less. This is the histological definition of arteriole. Adventitia is also very thin. So we define arteriole by observing the number of layers in the media histologically. You see, after that, gradually the muscle coat reduces its size, the layers reduces. So when it becomes one continuous layer, it's still arteriole. But when this only layer becomes rarefied, smooth muscle separates from one another. This is known as metarteriole. So a discontinuous one layer of smooth muscle remains in the arterial wall. At the end, this smooth muscle makes a continuous ring, single smooth muscle ring. This is known as pre-capillary sphincter, because pre means before, because after that capillary starts. So we'll get a large artery becoming medium artery, becoming smaller artery and much smaller, and ultimately when it contains 2 layers of smooth muscle in the media, it is arteriole. Again the layers become thinner and when the media contains single discontinuous smooth muscle layer, this is meta-arteriole, then the smooth muscle makes a continuous ring, a single ring. This is precapillary sphincter, and after that capillary starts. You look at this, this is meta-arteriole because it is continuous epithelium, this is arteriole, and this one is the precapillary sphincter. You can see the ring, and after that this is the capillary. This is arteriole, single continuous layer of smooth muscle is in the wall. This is arteriole, you can see one or two layers of smooth muscles are there. So arteriole has one or two layers of smooth muscle. This is the histological classification. If we see by naked eye, it's very tough to see. Magnifying glass can be used. If it is less than 0.1 millimeter, this is arteriole. So gross classification of the arteriole is the artery having size, caliber, diameter less than 1 millimeter size. Meta-arteriole, we can define this is discontinuous single layer of smooth muscle in the wall, then precapillary sphincter, and then capillary is formed. You can see this is the precapillary sphincter. Here is the precapillary sphincter, then capillary starts. This is the capillary bed. This one is the arteriole, then meta-arteriole and capillary beds. This one is the artery because more than one layer, two layers you can see. These arterioles are regulating the flow of blood in the capillaries. So arteriole can contract and can reduce flow of blood to the capillary. So arterial blood pressure is maintained by the arteriole. If all the arterioles are open, then diastolic blood pressure. Thank you very much for remaining with the presentation.